Oh, there you are. Okay. Oh, hello. I'm Mabel Fuentes Vergara, and I'm a PhD student at Dr. Kulati Lab at Queen's University, where we study invasive species. And today I'm going to share some of the research done in the quest of identify candidate genes that may be involved in the flavonoid pathways of Lythrum salicaria to study hypothesis of invasion success. Climate change is exacerbating environmental stress, leading to rapid evolutionary adaptations in species. Introduced species are present in biogeographic regions where they did not evolve and to which they might be poorly adapted, encountering a suite of novel stresses and selection pressures. Introduced populations have higher potential for rapid adaptation, which can explain why some introduced species establish, proliferate, and become invasive in new environments. There are several hypotheses to explain plant invasion success, and here are some of them. While these hypotheses cover various aspects of biotic interactions and resource dynamics, they do not fully integrate the role of abiotic stressors like temperature, UV, or soil salinity or contamination. I'm not sure if there's any integrated hypothesis that cover every aspect, but to test this hypothesis, we need to examine both metabolic and gene expression of these plants and focusing on their responses to biotic and abiotic factors. This includes studying how plants relocate resources from defense to grow and adapt their defense mechanisms, and potentially produce unique biochemical compounds. Additionally, we would need to analyze how invasive plants respond to fluctuating resources and abiotic stressors like temperature, water availability, and nutrient levels. Assessing the gene expression changes related to stress response, resource efficiency, and adaptations to these biotic stressors. So my PhD thesis aimed to investigate how genetic and molecular mechanisms influence the rate of adaptation in response to environmental stressors. Specifically, I seek to understand how molecular level adaptations translate into functional responses that enable survival under various environmental stresses and how selective pressure from these stresses shapes genomes and gene expression during the invasion process. I'm on the early stages of my research, but I'm going to present the exploratory data that I've gathered so far. For that, now I'm going to introduce our study system. This is Lythrum salicaria, also known as purple loosestrife. It's a perennial wetland plant native to Eurasia, where it has been historically valued for its medicinal properties. But in North America, it's considered invasive due to its rapid spread. Although this species doesn't seem to threaten the diversity of density of other wetland plants, it has the potential to become dominant due to its size and perennial nature. Its capacity for rapid adaptation to new environmental conditions allow purple loosestrife to naturally spread around Eurasia. The time of arrival to North America is uncertain, but the oldest records date to early 1800s. The most likely explanation for its introduction, it was purposely brought here as a medicinal plant and for its ornamental qualities, because it's very popular among beekeepers. This plant has rapidly evolved differences in flowering time to adapt to differences in season length. In northern regions, flowering early in the growing season allows them to produce more flowers before the first frost. But in southern regions, it's better to wait and grow bigger before flowering. This adaptation is genetic in nature because if we grow seeds from plants from different latitudes under the same conditions, they will maintain this pattern of flowering time. This plant can grow under a variety of environmental conditions and can even tolerate soils with heavy metal contamination. Lithium salicaria contain a wide range of chemicals, including alkaloids, tannins, anthocyanins, glycosides, terpenes, esteroids, organic acids, phenolic acids, and flavonoids. This arsenal of secondary metabolites offer protection against abiotic and biotic stressors like herbivores and pathogens. These compounds also help attract pollinators and predatory wasps that eat the herbivores. So among all those compounds, I choose to study flavonoid metabolism because flavonoids play diverse roles, including acting as antioxidants, UV protectant, and defense compounds against herbivores and pathogens. They also influence pigmentation, pollination attraction, hormonal signaling, and also regulate flowering time. Additionally, studying evolution of flavonoid biosynthesis genes provides insights in the broader evolution of metabolic pathways, revealing how plants adapt their biochemistry to diverse challenges and stresses in new environments. Here is our general flavonoid biosynthetic pathway based on the distribution patterns of the flavonoid subclasses. It has been suggested that these pathways may have evolved via a series of steps, and that the first flavonoid biosynthetic enzymes were chalcone synthase, chalcone isomerase, and flavanone 3 hydroxylase. From that, the pathway branches to produce isoflavones, flavonols, dihydroflavonols, and flavones, and also anthocyanidins and anthocyanidins, which confer color to flowers, among other functions. And here is a list of flavonoids I found in Lithium salicaria that are reported in the literature that I have been studying for medicinal purposes. Most of the identified compounds are derivated from the flavones luteolin and apigenin, but many had not been characterized. None of the enzymes in the flavonoid metabolic pathways have been identified or characterized either, which motivated us to explore the genetic of these pathways and by analyzing the transcriptome of some of the plants growing in a common garden experiment. In 2017, a former lab member established a common garden experiment at Queen's University Biological Station using collected seeds from wild populations of Lithium salicaria from eastern North America, and the plants have been monitored for several years now. As part of my master's thesis, I assembled the transcriptome of inflorescence from Lithium salicaria plants growing in the common garden. We collected tissue samples from 4 plants derived from a single population near Hancock, New York, to analyze differences in flowering time gene expression. The transcriptome was assembled using a draft genome of the diploid individual from the native range as a reference. RNA was extracted from the 4 plants and 4 tissues per plant with a flowering date difference of 10 days, which we call them early and late flowering. RNA was sequenced using Illuminature reads. Not all the samples made it to the end. The RNA extraction process resulted very difficult due to all the polysaccharides and other compounds in the samples. I assembled the transcriptome of 10 samples using the STAR and string tie 2 programs. The assembly resulted in 120.5 thousand transcripts. I did a differential expression analysis also of the transcript with different tissues and flowering time as factors in the DI-seq model. On the left here is a volcano plot of one of the DI-seq models using the up-flame trinket method. Labeled transcripts are some of the most up and down regulated transcripts, and the heat map on the right shows the 20 most differentially expressed transcripts, clustered by flowering time rather than tissue. I aligned the translated sequences from the longest open reading frames using the BLAST-P to a UniProt subdate database of 299 orthologs from 100 plant species, consisting of proteins related to the flavonoid metabolic pathways. Over 3,800 unique transcripts significantly match 170 orthologs from 61 species, with 1,079 transcripts strongly matching 77 proteins from 48 species. The percent identity ranged from 34 to 99%, indicating both highly divergent and conserved sequence, respectively. These results indicate that they may be a high diversity coming from standing genetic variation. Here is a more extensive flavonoid biosynthetic network of some of the proteins that have matches, which are highlighted in green. More specialized metabolic enzymes are not included in this pathway. And these are the sequences that are corresponding to the transcript that are upregulated in early flowering plants. The differential expression of these transcripts suggests a possible functional role in flowering time, but further confirmation is needed since the sample size was too small to conclude anything. And since there's evidence of the chalcone synthase, the first enzyme of the pathway, having a role in controlling some of the circadian flowering time pathways, is worth exploring this transcript in more detail. And here is a phylogram of alignment using cluster of 8 sequences highlighted in blue of predicted proteins with more than 70% sequence similarity with chalcone synthase, and blast P value of less than 1 over minus 100 from orthologs of the UniProt database. In red, there's a predicted protein from a differential express transcript and is upregulated in late flowering time, flowering plants. And since I ran out of time, this is all I can share today. For my intention for my PhD is to seek identify and characterize genes and metabolic pathways associated with rapid adaptation of flower phenology and their modulation by stress responses, examine the role of polyploidy and chromosome structure in the evolution of these genes, and establish links between phenotypic responses and genetic biochemical pathways. So, and additionally, I will explore the trade-offs and interactions between biotic and abiotic stress responses. Thereby, they contribute to how salicaria rapidly adapt to various environmental challenges. So the completion of this work wouldn't be possible without the contribution and support of many people. So thank you all for listening. Do you mean by each one of them or group them? Yes, I did several analyses, and I analyzed the Go terms that go with the transcripts. They were very different and all over the place, so yeah, thank you.
I'll go ahead and get started. Hi, everybody. My name is Andy Lee. I'm a PhD candidate working with Mark Christie at Purdue University. Today, I'm really excited to talk to you a little bit about the transcriptomic work that we've been doing on Kellis whelks, which is pictured here on the right. Just a little bit about the whelks. They are a kelp forest gastropod found in the Eastern Pacific. So they range all the way from Baja California in Mexico to Monterey in Central California. They share many life history traits with other ecological and economically important species. So they have these relatively sedentary adults that lay eggs. So eggs are in these capsules that lay egg on rocky substrate. And within these capsules, the larvae mature from the trochophore to the villager stage. So that kind of turn from a white to a gray. And once they reach the villager stage, they are released into the water column where they spend 40 to 60 days dispersing with ocean currents. And this pelagic larval stage is shared with many, many marine species and affect a lot of ecological and evolutionary processes of marine organisms. And so after that pelagic larval stage, they will metamorphose and settle. And so that pelagic larval stage, that dispersal kind of affects a lot of what I'm going to be talking about today. And if you've been to a lot of marine genomics talk, this is perhaps not surprising, but marine populations are hard to resolve, especially at neutral loci. So here is an example using spiny lobsters, looking at structure plot, and across these 9 sites, we don't see any population structure. Same thing on the PCA, so each color and shape represents a different site, and we also don't see any population structure in the polar cod. Kellett's whelks, like many marine species, have expanded their biogeographic range in response to warming sea temperatures. So they move past a biogeographic break at point conception in Southern California in the dashed line. So now they're in the expanded range. So anything above that is their expanded range, and anything below that is their historical range. And there are novel conditions that the whelks experience in the expanded range. And for example, they experience colder water. So on the bottom left there, I'm showing sea surface temperature over our collection period. And so note the black line, which represents the Monterey population, is much colder than the historical range. So using samples we raised in the Carmen Garden from these 3 sites, we previously found population structure across both the transcriptome and on differential expressed genes that we call between the expanded and historical ranges. So here, our PCAs show population structure. Same thing with structure plots where we saw population structure at k equals 2, which had received the most support. We also see population structure in neighbor joining tree, it's kind of just showing you the genetic relationships between each of our samples. And we saw population structure both in the differential expressed genes and sort of SNPs across the transcriptome. So the SNP with both the highest predictive importance value and amongst the highest FST occurs on the gene triose phosphate isomerase. So here on the y-axis is importance in a random forest model that we trained to predict a site of origin for a sample, and then x-axis is pairwise FST between the expanded and historical range. The orange are differential expressed genes, and the black are the SNPs across the transcriptome. So TBI is the top right. It's got one of the highest FST values and the highest importance value. It's a gene that's involved in the metabolic processes, both glycolysis and glycogenesis, and it's also implicated in cold stress response. TPI was both highly differentiated, so the left graph here shows just allele frequency of all the SNPs that occurs on TPI, and it was differentially expressed. So this is the normalized gene count of TPI in the historical and expanded range. So in this study, we sort of identify population structure across the transcriptome and on the differential expressed genes, and some of these differential expressed genes are and the expressed genes, I guess, are related to metabolism and cold tolerance. Because we found population structure in both the DEGs and across the transcriptome, ostensibly some of those SNPs occur in regulatory regions of the differential expressed genes, and those SNPs may represent the genetic basis for population differences in plasticity. So they're adapting to cold temperatures, and that's kind of cool. But the oceans are warming, perhaps not super surprising. So globally, the ocean temperatures are rising, and locally as well, so both in the long term, in the short term and the long term. So we found evidence that the whelks are adapting to colder temperatures in the expanded range, and now we're asking, can Kellett's whelks adapt to warming ocean temperatures as well? So to address these questions, we collected adult whelks from 4 different sites across the whelks range, and these sites had different thermal conditions. So on the bottom left, I'm showing you the sea surface temperature plots again across 2021. So we collected wild samples and we acclimated them, adults in a common garden. We then collected offspring egg capsules when they are laid. And once they're laid, we take the capsules and move them and expose them to 2 different treatments. One is controlled conditions, which is just ambient temperature in our common garden, and a heated treatment, which is plus 4 degrees Celsius. So these larvae spend their time in these treatments until they're mature to the villager stage. We then conduct RNA-seq on those F1 offsprings at the villager stage right before they're released into the water column. It's kind of a different way of looking at the experimental design. So to recap, we got wild-caught adults from 4 sites, acclimated them in a common garden, and then exposed them to different experimental temperatures, and then conducted RNA-seq. We then identified differential expressed genes between the control and heated treatments in each of the 4 sites using DSeq2, and then below is the result of those differential expressed genes. I want to know the various number of differential expressed genes among sites, and particularly there was fewer in Naples NAP. So to explore this relationship further, I visualized the number of DEGs on the map. So each dot here represents unique DEGs. The size of the dot represents a unique number of unique DEGs, and each line represents shared DEGs between 2 sites. So a couple of interesting things here. Although they're the furthest oceanographic distance apart, Monterey and Point Loma share really similar expression profiles. And then even though they're closest together, Diablo Canyon and Naples are very different. And then because there's a biogeographic break and Point Conception, we also compare the expanded range populations, and they seem to be more like each other than the historical range populations do. And I think a lot of this pattern can be explained by the fact that Naples just had a lot less differential expressed genes when exposed to the elevated temperature. So the last slide was just pairwise comparisons between each site, and I also wanted to look at other relationship as well. So this is just a histogram of number of DEGs and how many times they occurred in a site. So while most DEGs occurred on just 1 site, there were also DEGs that were shared among sites, some of them within range, some among range, and a handful of DEGs that represent a shared response across all 4 populations, and that'll be the focus of the rest of the talk. We wanted to explore the function of these DEGs that were shared among the 4 sites, and so we used annotation using eggnog mapper, and then to identify the genes, and then we conducted gene ontology enrichment. So here we're comparing the gene ontology terms associated with those 124 differential expressed genes with the genome that we recently assembled of Kellett's whelks, so to see which terms are enriched. So goal terms are usually really hard to understand, so I tried to simplify the terms here a little bit. This plot shows only the very, very significantly enriched terms as terminal nodes, and then it kind of visualizes the relationship between each goal term. So the central term there is biological process, and each of the terminal terms are a goal term that was significantly enriched in the globally expressed DEGs. And each color represents a subcategory of goal terms. So on the left side of this map, I guess, here, we see like usual suspects when you look at goal terms, so like cellular process, metabolic process, and biological regulation of metabolic processes as well, so those terms, which we see often. And so I wanted to zoom in a little bit and look at some of the other terms that we see in our DEGs. First, among the global DEGs, we see a lot of goal terms associated with stress response, which is perhaps not surprising because we expose them to some thermal stress. So that's cool. Zooming in a little bit, we also see terms associated with development, and that included determinations of adult lifespan and aging. We also see terms associated with reproduction, including developmental process involved in reproduction. So again, with the development of these egg caps inside these egg capsules. So to conclude, we found varied expression profiles across the Kellis-Wilks biogeographic range, where in response to elevated temperatures. The global shared differential expressed genes are associated with reproduction, development, and response to stimulus. And this is sort of like in the preliminary stage of analysis. I still have a lot more to explore. So today we only look at the smallest bar, and I have to look at all the other differential expressed genes as well. So I'd be happy to chat about other possible analyses that folks might have with a data set like this. We are also interested in looking at constitutive expression, so genes that are expressed both in the ambient treatment and in the elevated temperature treatment. And then we're also going to do WGCNA, which kind of look at co-expressed gene networks within the 4 different sites. And lastly, since it's a symposium on also dispersal, we would love to know about the population connectivity between these populations. So here, we're wondering how connected these populations are the historical and expanded range connected, and if the historical samples are more adapted to warm temperatures, are they being transported into the expanded range? So more of that to come. With that, I'd like to thank my funders, my collaborators, and my lab. This work cannot be done without a bunch of other people. And yeah, I'll take any questions. We did try to do that in the paper. I didn't get to talk about it today. We did a Monterey and Naples cross, but it would be great if we could know the sex of the snails before we cross them, and they don't like to mate when we put 1 and 1 together. So we actually don't know who mated with who within that cross. So there's a little bit of sort of exploratory work to be done there, and I'd love to chat more about that. Kind of confusing.
Hi, my name is Alison Hall. I'm a PhD candidate in the Pespini lab at University of Vermont, and I'm excited to share with you some research from my grad school thus far. So this is kind of my favorite way to look at how dramatically temperatures are rising in the ocean. In this graph, you can see daily sea surface temperature in the North Atlantic plotted over the course of a year. And in orange, you can see that 2023 was a really remarkably warm year. And in the solid black line, you can see that 2024 is also on track to be really remarkably warm. And in addition to these increasing average warm temperatures that we're seeing, we're also seeing an increased frequency of warm anomalous events in the ocean. In this graph from July of 2023, you can see that the darker and more bright red colors are more warm departures from the average. And you can see here there's a lot of 3s and 4s, which represents 3 and 4 degrees above average, which might not seem that intense of a warming day for us, but it can be quite impactful for organisms. So the central question which I look at in my dissertation research is, how do animals respond to these warmer conditions? And we often think about response to environmental change in 3 broad paradigms. We think of how an organism might stay and adapt or acclimate to their environmental conditions, whether they might move to find a new habitat, or whether they're unable to survive in their current habitat. And my work focuses on adaptation and acclimation, and specifically today I'll be talking about acclimation and specifically phenotypic plasticity, which I think about as a change in a phenotype in response to environmental change without an underlying change in genetics. And in particular today we'll be talking about developmental plasticity of the upper lethal temperature trait in the copepod Acarchatonsa. And we really like Acarchatonsa because they're broadly distributed, they have pretty short generation times of about 21 days, and they're really ecologically important. They're kind of that animal base of the coastal food web. Additionally, they exhibit this really remarkable developmental plasticity of their upper lethal temperature trait. And in these data, in blue, we see animals that are raised at 18 degrees Celsius, and in pink, we see animals that are raised at 22 degrees Celsius for just one generation. And we see that being raised at 22 degrees Celsius increases the upper lethal temperature by about 1.5 degrees Celsius. And these data are collected by putting 2 copepods in a glass tube and then ramping up the temperature slowly and recording the temperature at which they die. So it's a good way of understanding how much heat they can take before they die. So what we'll be discussing today is what are the molecular underpinnings of this developmental plasticity that we observe. And to address this question, I collected copepods from Cape May, New Jersey, and I brought them back to Vermont, where I reared them for 3 generations at 18 degrees in a common garden in the lab. And then I split the 3rd generation's brood into two different developmental temperature treatments, where they were raised for 1 generation at either 18 degrees Celsius or 22 degrees Celsius. And then the adults from these broods were exerted to a 1-hour sublethal temperature exposure, where if they didn't experience the temperature exposure, they just remained at their developmental temperatures for an hour. And if they did go through that sublethal temperature exposure, they either went through it at 28 degrees Celsius for an hour or 33 degrees Celsius for an hour. And then I extracted RNA from those samples to look at gene expression response to both developmental temperature as well as acute temperature exposure. And I aligned those RNA sequence data to the Acarchatonsa reference genome and quantified reads using salmon, and here they are visualized as a PCA. So here the shapes represent developmental temperature. So the circles represent animals that were developed at 18 degrees Celsius, and the diamonds represent animals that were developed at 22 degrees Celsius. And the colors in this PCA are the final temperature. So base refers to baseline, so either 18 degrees or 22 degrees. Orange are animals that went through that 1-hour exposure to 28 degrees Celsius. And 28 degrees Celsius represents kind of a warm day in their current environment. And in red, we see data from animals that went through 33-degree exposure, and 33 degrees is kind of a warm future condition day in their current environment. And what hopefully you can see in this PCA are strong effects of both developmental temperature along PC1 in the shapes, and effects of final temperature along PC2 represented in those colors. And then digging into these data a little bit more, we can look at differential gene expression between our treatments. So we'll start off by looking at differential gene expression between our 2 developmental temperature treatment, within our 2 developmental temperature treatment lines. So here in orange, we're looking at baseline, so either 18 or 22, and differentially expressed genes between baseline and 28. And in pink, we're looking at differentially expressed genes between baseline and 33 degrees. And using this approach, we can ask the question, how does acute heat exposure affect gene expression? And in these data from our animals developed at 18 degrees Celsius, we see a really large gene expression response between 18 degrees and 33 degrees. And then a more muted response between 18 degrees and 28 degrees. And looking at our animals developed at 22 degrees Celsius, we can see that that pattern pretty much holds, but there's a much larger magnitude of differentially expressed genes when the animals develop at 22 degrees. So overall, we saw a stronger response to acute temperature change when copepods developed at a higher temperature. And digging into what these genes were that were differentially expressed, we see that genes involved in heat shock are differentially expressed in response to acute temperature exposure in both developmental temperature treatment groups. So what this means is that these three genes, HSP90, HSP70, and heat shock beta, which are all involved in the heat shock response, were differentially expressed in both of our developmental treatment lines. But as you can see in this graph, the developmental treatment lines, these genes weren't differentially expressed within those. So what that tells me is these genes are important to acute temperature exposure response in both of our developmental temperature treatments, which makes sense, it's heat shock. So next up, we can compare between our two developmental temperatures to look at, does expression at acute temperatures differ between developmental temperature treatment groups? So in this next Venn diagram, the green circle will be comparing differentially expressed genes between animals developed at 18 and 22 that did not go through a heat shock, high heat exposure. 28 degrees will be in orange, and 33 degrees will be in pink. And what we see here is a really large number of differentially expressed genes at baseline, and then a slightly smaller when we compare between the developmental temperature treatment groups at 28, and then a much more shared response to exposure at 33 degrees Celsius. So overall, we see major differences at baseline conditions, represented by that large number at baseline, and then a largely shared response to acute exposure. And digging into the functionality of these genes a little bit more, we see that upregulated at baseline between our developmental temperatures are genes involved in RNA processing, gene regulation, and DNA repair. And downregulated at 22 in comparison to 18 are genes involved in developmental and anatomical processes. And then looking at these 6 genes which responded to both acute temperatures differentially between our two developmental temperature treatment lines, we see genes involved in mitochondrial processing, indicating that those genes might kind of start to explain that plasticity that we see at development at 22. And then using WGCNA analysis, we can look at whether there are co-expressed gene modules that correlate with upper lethal temperature trait. And we see this dark turquoise module is highly correlated with our upper lethal temperature, and genes in that module are involved in mitochondrial functioning, specifically involving membrane transport and metabolism, indicating that those genes are really important to our upper lethal temperature trait. So to summarize, we learned that copepods that develop under warmer temperatures mount a larger gene expression response to increased temperatures, and that baseline gene expression response is highly differentiated between developmental temperature treatment groups, but that response to acute temperature increase is largely shared. And genes involved in mitochondrial function underlie differential expression response to acute exposure and upper lethal temperature response. So to go back to our original question, what are the molecular underpinnings of developmental plasticity? We learned that gene expression priming at 22 degrees Celsius, specifically in genes related to RNA processing and DNA repair, and expression of genes related to mitochondrial function is positively correlated to temperature tolerance. And then to quote Andy, this is a symposium that includes dispersal. So Matt Sasaki revealed that there's a genetic component to this developmental plasticity that we see along this latitudinal cline. So we see at more northern latitudes, animals have a lower thermal tolerance, but a higher developmental plasticity. And at more southern latitudes, animals have a higher thermal tolerance, but a lower developmental plasticity. And so in this species with low dispersal capacity, these mechanisms that we talked about today of developmental plasticity may be important in coping with a warmer and more variable future, which might actually put those animals in the south with a lower developmental plasticity capacity at a greater risk. Thank you so much for listening. Thank you especially to Manali and Melissa and the Pespini Lab and my funding sources. And if anyone has any questions, I'd be happy to answer them. Sweet.
No, just very quickly. You were using wrapping acids, right? Did you try slow versus fast wrapping acids? Because it supposedly plays a victim in the ability for them to respond.
Oh, yeah. Yeah, so the question was, did I experiment with different timing for ramping assays? This assay is largely based on a previous PhD student in the lab, but personally I played around with whether it's like amount of time at a warm temperature. So for example, 33 degrees is like fairly close to the population that I did this pilot in. And I had them at 33 degrees for like 6 hours, and there was no lethality observed. So that kind of made me think that it was more the temperature than like the duration of time. Yeah, but something good to play around with, because I'm sure there's a huge effect.
All right, good afternoon, everyone. Thank you so much for being here today. My name is Moisés Bernal, and I'm an assistant professor at Auburn University. And we're trying to understand how marine fishes are responding to a changing planet. And in this case, I'll tell you a little bit of the research that was done by Ali Swank, a former student in the lab, Erin Chille, and Bridie, that are collaborators from University of Otago in New Zealand. So first of all, I hope I don't have to convince anyone here in this room that the climate is changing. And because of these changes, we're going to have an increase in ocean temperatures. And the question is not so much what is going on, if it's going to be warmer or not. The question is, what is the degree of warming and how quickly it's going to happen? And in order to do that, there's estimates of the emission trajectories, but now there's also an estimate of the socioeconomic pathways. So if we collaborate as different countries, there's going to be a different response than if everyone relies a lot on fuel and there's constant wars between different countries. Not only is the average temperature going to change, as probably everyone here knows, but also marine heat waves have become much more intense and much more frequent, especially over the last 30 years. And you can see that there's an example here from the summer of 2024 where heat waves have been affecting ecosystems across the world. Last year, there was a very intense heat wave that affected coral reefs all over the world as well. And it's not only the average temperatures that are going to increase, but it's also the increase in the intensity and the frequency of the heat waves. And this is going to be a very, very worrisome scenario for organisms that do not regulate their internal body temperature. Most marine organisms are actually ectotherms, so they depend really heavily on the external environment for getting cues about when to develop, when to eat, when to move, and how to do all these processes. So temperature is a very big determinant for them in what they're doing in their everyday lives. So in the case of coral reef fish, when you actually have an increase in temperature, warming is going to lead to a very big increase in aerobic demands. And also, at the same time, while this increase in aerobic demand is happening, in the case of coral reef fishes, their ecosystem is also going to be affected because warming will increase the propensity for bleaching and also the coral reef mortality. So what we're actually seeing is that in several sites where you have massive coral bleaching followed by mortality, what's going to happen is that over time there's going to be very substantial erosion of that ecosystem. So you go from a heavily complex ecosystem with a very, with lots of branches and a very complex three-dimensional structure to kind of a flattened out environment that will change the dynamics amongst organisms. So if you think about it, this creates kind of a problem for marine fishes. On one side, they're changing their aerobic demands and the metabolic demands because of warming. And then on the other side, their ecosystems are becoming heavily affected. And we're not the first group to actually ask these questions. So Morgan Pratchett has been very, very effective at actually measuring how coral reef fish communities can be affected. And there's a very intense effect before and after bleaching in terms of the fish that actually can live in those more simple ecosystems and those more simple environments. So the three-dimensional structure actually can have an effect on the presence and absence of fishes. But in this case, we want to ask in like a generalist fish that has very high plasticity, what's actually going on in their brain and in their behavior to try to understand how fish are responding to this double threat of warming and habitat simplification. So most of this work was done by Ali Swank. She's no longer with us. She's still alive, but she's a PhD student in Sarah Davis' lab at BU. And the idea is that for 10 days, we subject this damselfish right here, Abudephtus saxatilis, to different combinations of warming and complexity. So 27 degrees Celsius is kind of a control average temperature for the Gulf of Mexico. 31 represents a measured marine heat wave event. And then we had high complexity right here, all these fake plants and cups and different PVC pipes, and the low complexity, a very sad fish in an empty tank. And here we measured differential gene expression in the brain, and we also wanted to see how it affected oxidative stress in the liver. Right off the bat, what we see is that the biggest effect was actually temperature. So the PCAs actually explain mostly temperature, and we didn't see a very intense effect of habitat complexity in terms of differential expressed genes. So only looking at habitat, we didn't see any genes that were only affected by simplification of the habitat, but we did find 285 differential expressed genes that were associated with temperature. And that included the activation of heat shock proteins. And also it led to downregulation of things related with DNA damage, tissue regeneration, and also things related with the regeneration of the central system. So we can see that temperature is actually leading to a very intense effect to the actually ability to repair the nervous system. When we look at the GO categories, we see that there's actually deactivation of processes that are really important for the transmission of neuronal impulses. So things related with ion transport, ion channel activity, and so forth, and the voltage-gated channel activity, they were all deactivated. And this is super important for the recovery and the maintenance of these sort of neuronal impulses. And in a previous paper, we've actually found similar responses in a transgenerational experiment for another damselfish. And there's also behavioral studies that suggest that when you have very intense warming, there's actually uncoordinated behavior. There's not as good as a response in terms of feeding, and the fish become less efficient at problem solving whenever there's warming. So we're seeing this from both a molecular and a behavioral perspective. Another thing that was interesting is that when you add temperature, and so here we are comparing low complexity control versus heat wave, and here we have high complexity control versus heat wave. In all the comparisons related with low complexity, there was a synergistic effect between complexity and warming, where fish exposed to low complexity and warming actually had an order of magnitude higher response in terms of differential gene expression, which means that when you have, even though the complexity on itself did not give us differential expressed genes, when you have the combination of both, the response is actually magnified, which is a very interesting result. So after that, we also saw the analysis of oxidation, and we see that in heat wave temperatures that the fish are experiencing today, they actually get the accumulation of these sort of oxidative damaging proteins in the liver of the fish, which is pretty worrisome. So after that, for Ali's master's, we actually did this study in collaboration with these two magnificent undergrads who spent a lot of time actually looking at these videos. And the idea was to grab fish that were about 2 or 3 weeks old in some cases, and they were experiencing high complexity and low complexity for 3 months. And then after that, Ali, the undergrads, did this amazing assays to test for aggression and boldness, and all these were recorded in video. So there was no significant difference in the size of the fish, and this gives you an idea. In the aquarium, here's a fish looking outside of the window, kind of sad, playing some emo music in the emo corner. And here's a much more active, vibing first floor where like it's just really nice apartments and they have a lot of vegetation and so forth. And the idea was that with this, Ali here seen monitoring all the fish. The idea was to see the amount of time that they were spending in front of the mirror, the amount of time that they spent attacking the mirror, and also if they had any sort of displays, such as biting, trying to interact with the mirror, or have a lateral display with the mirror. So you can see here in the video the number of bites that there was an increase in the number of bites for fish that were actually in the simplified environment. So where they're attacking the mirror at a higher frequency than the fish in the high complexity. The low complexity fish were also doing more lateral displays than the ones, so they were much more aggressive towards the fish in the mirror. And then they also had much more aggression in terms of frontal interaction. So these means that there was much more higher aggressiveness in the fish from the low complexity environment. After that, we wanted to see the interactions with a novel object, so we have to find a heavy bottom object that it wouldn't topple with the pumps in the aquarium. This Patrick star was a perfect candidate for it. And the idea was to see how much time the fish were actually observed interacting with the new structure. And you can see here that there's some fish that actually had exploratory behavior, that they were kind of swimming around this novel figurine, or some of them were so scared that they would just be in the corner. So scoring the exploratory behavior, Ali and the two undergraduate students actually found that fish in simplified environments actually had a much higher rate of exploratory behavior compared to the high complexity fish. So it's not only differences in expression, but also differences in their behavior. So now we're trying to actually expand these questions, not just in coral reef fish, but also in more temperate fish. And then this is a work that we're doing in collaboration with Eleanor and Bridie, where we're trying to understand what are the responses of habitat simplification in the common triple fin. So they actually ran for 1 year this experiment where they took juveniles that were between 2 and 3 months of age across 20 mesocosms per treatment for 1 year of either simplified environment where they just had very small PVC pipes to fight over, or this very complex ecosystems where they were large rocks and also this pots, clay pots where they could hide. And what we actually see firsthand is that there was actually differential survival between the two treatments, where the complex environment actually had a significantly lower mortality rate than the simple environment. So about 40% of the fish in the simplified environment died, and we think that's because there was so much competition for the available space that they were not able to sustain that for a long time. And then when we look at the differential gene expression of the brain, we actually see that the samples, if you kind of squint, you do see that there's kind of difference between complex and simple. Again, this is just habitat complexity. This is not related with temperature. And what we can see here is that we couldn't really disentangle effectively the effects of habitat and sex, but when we compare complex versus simple environments, we get 92 differential expressed genes, and several of those categories suggest that in complex habitats, we get activation of protein synthesis, we get effective formation of myelin protective sheets. We also have optimized gas transport, and also the biosynthetic processes for control of inflammation and oxidative damage. Whereas in simplified habitat, we actually see increased of adipose tissue development and a lot of categories that are related with transporting activity and synthesis and metabolism of vitamin B12. So there's something there where the interactions that they're having in that simplified environment is actually leading to differences in how they're transmitting these different neurological impulses. So in the end, in warmer, simplified habitats, it looks like we're going to have differential expression in terms of neurotransmission, immune responses, oxygen transport, the protein synthesis, and also tissue repair. We can see increased activity, increased aggression, and boldness in simplified environments. And then we're going to have differences in survival, not only based on the competition, but also in the more bold behavior that fish may have. And then on top of that, we are starting to see that there's synergistic effects between warming and habitat simplification that might affect fish in the future. And one of these papers already went out. The other one hopefully will be ready soon. And then the one with Eleanor Kelly is still in preparation. So with that, I just want to thank the funding sources, lab members, and thank you for your attention. Yeah.
So the essay you did on the fish, on the environmental complexity, you only had one fish per tank.
It was one fish per tank.
Do you know if, like, when you put multiple fish, if it changed completely the behavior, or?
With this fish, it would be a bad idea because they're very territorial, so they kill each other. Maybe they'll kill each other faster if there's no environment. That would be my prediction. But yeah. Yeah, I'll take any other questions in the poster session. Thank you.
Oh, there we go. Hello, everyone. My name is Erin Chille. I'm a PhD candidate at Rutgers University in the Bhattacharya Lab. And today I'm thrilled to be talking today about about how we found out that gene expression response in two reef-building corals is primarily driven by ploidy and genotype. So first, some background. As you can see, coral bleaching has been making the news a lot in 2024. But what is coral bleaching and why do we care? Well, first, I have to tell you a little bit about what corals are. You probably know that, or are familiar with corals in terms of when you go on vacations, you see these beautiful coral reef assemblages with lots of fish. But these coral reef assemblages can be broken down to smaller units. For example, these 3D coral colonies provide the structure that accumulate and attract the reef fishes. And each of these coral colonies is made up of hundreds of polyps that are very jellyfish or anemone-like, and they're all clonal groups. And what you may not know is that corals actually exist as a holobiont. That is an association of animals and other organisms that exists with the coral animal and its associated microbiome, which includes eukaryotic and prokaryotic microbial community. And the most important member of this microbial community is the symbiotic dinoflagellates known as Symbioneciae that are the foundation for the coral reef ecosystem by providing nutrients and photosynthesis in exchange for a safe habitat inside the coral tissues. And these exchange of nutrients allow corals to biomineralize that create the 3D structures of the reef. So what is coral bleaching? Coral bleaching is essentially the dysbiosis between the coral animal and its algal endosymbiont. And as you can see on the far right, you have a beautiful pigmented coral animal, and as it gets more and more stressed, it becomes a ghostly white, and this is because of the expulsion of the algae from the coral's tissues. And increased thermal stress, especially from more frequent and intense marine heat waves, are the most common cause of coral bleaching. And why do we care, you may be asking? Well, as I said, corals are ecosystem engineers that are foundations for the coral reef ecosystem. They provide biodiversity hotspots that account for about 20% of marine biodiversity, despite only accumulating about 1% of the seafloor. And their ecosystem services support millions of people worldwide in terms of tourism, reef fisheries, and shoreline protection. So, as you may be guessing, coral reef research is largely focused, or at least in part, on understanding the genomic basis of bleaching and adaptive capacity of corals to thermal stress. And transcriptome data has been a powerful tool to understand bleaching and adaptive capacity in corals. So we set up an experiment to ask, what are the transcriptomic markers that are indicative of tolerant and non-tolerant phenotypes in a natural coral population? And this is unique to many other coral experiments because many other coral experiments are based in only one or a few genotypes. So we took advantage of two large natural populations of corals in Kaneohe Bay, Hawaii. So we collected about 400 specimens, one sample from each coral, from about 400 coral colonies of the stress-sensitive Passalopora acuta and the more stress-resistant Montipora capitata. We took some samples for physiology and some samples for gene expression, and we exposed them to 8 weeks of temperature stress. And the assumptions underlying this experiment were that each colony would represent a genetically distinct individual, that all colonies would be diploid, and that gene expression variation would be driven by treatment. And the outcome of this experiment was expected to reveal a core set of stress-responsive genes shared between the two species and populations. So, how did they respond? Well, this actually is not my project, so I'm just going to give a little bit of a primer so that we can get more into the gene expression stuff. But I encourage you, if you're interested, to click the link to go towards this publication to learn more. So postdoc Emma Strand did the majority of this physiology work. And what she found is that, as expected, Montipora was more thermally resistant than Passalopora, according to a bunch of physiological metrics, including color score, chlorophyll content, symbiont cell density, and antioxidant capacity. She did find that both species showed a significant physiological response to stress, and that the lower performance of Passalopora acuta could be primarily accounted for by lower antioxidant capacity and elevated baseline photosynthetic rates. So this allows us to inform our gene expression experiment. Based on this physiological experiment, we would assume that variation in gene expression should be driven by treatment, that shared treatment responsive genes should be part of the core thermal stress response in both populations, and that because Montipora had a lower physiological response to stress than Passalopora, that we expect a more muted expression response in Montipora than Passalopora. So, how did the gene expression respond to stress? And actually, we had some unexplained structure, especially in our Passalopora acuta, that did not correlate with any of our controlled or experimental variables. And Montipora capitata seemed to respond as we expected, with a little bit of variation explained by structure on PC2. So we went back to check the assumptions of our data. Seeing as gene expression variation didn't seem to be driven by treatment in Passalopora acuta, we went back to the data to see whether each colony represented a genetically distinct individual and whether all colonies were diploid. We called SNPs from the RNA-seq data in order to do this. And to check the underlying genetic structure, to check whether each colony represented a genetically distinct individual, we looked at pairwise relatedness, and to check whether the colonies were all diploid, we checked the density of allele frequencies that are informative of ploidy. So this is a heat map of pairwise relationships. I'm just explaining the heat map before I get into the actual data. This is a matrix of samples where each cell represents percentage of shared SNPs between two samples, and the bright yellow squares indicate a self-to-self relationship where we expect at greater than 94% of the SNPs to be shared between a clonal individual. So, as we see, check. Montipora appears to, each colony appeared to be a genetically distinct individual. However, something unexpected came up when we were looking at the Passalopora. Although we sampled 120 colonies for gene expression, we got only 8 genotypes. What the heck? So, each colony was not a genetically distinct individual. And if you look at the color barred indicated by the red arrows, it seems that there was no apparent isolation by distance, so even though there were only 8 genotypes that were found across the 120 Passalopora samples, they didn't appear to be structured by reef. So we think that maybe these individuals would be reproducing through parthenogenesis, and their propagules would be spreading throughout the bay randomly, depending on the currents. And then to check the ploidy, we have these density of allele frequencies, and again, I can't get too into it right now, but under our expectations for allele balance for diploid would be a normal distribution at 50%, and then 33 and 67 for triploid, and 25, half, and 75% for a tetraploid, et cetera, et cetera. So, looking back at our data, it looked like, yay, all Mantipora were diploid. They all had that perfect normal distribution peak at the 50% mark. However, oops, 60% about of our Pastylopora acuta were actually triploid. So this was very unexpected and had a major influence in how we went about analyzing our data henceforth. So, although Montipora met our assumptions underlying experiment, Pastylopora did not. Montipora, again, was an obligate outbreeder with all distinct genotypes and all diploid, but Pastylopora was primarily clonal, 60% triploid, and both fit the everything is everywhere model. So, going back to our original question, what transcriptomic markers are indicative of tolerant and non-tolerant phenotypes? We actually can't answer that with this data. So, we came up with some new questions. One, does gene expression reflect the physiological response to treatment, or is it more strongly influenced by genotype? And two, do genotype by treatment effects prevent us from detecting a core set of population-wide stress-responsive genes? So, going into this new first question, does gene expression reflect the physiological response to treatment, or is it more strongly influenced by genotype? And we used a PCA to test these two competing straw man hypotheses. We have these two hypotheses that represent the extremes in the data, and we don't actually expect the data to fit these extremes, but they help us understand where do our data most closely fit. So the PCAs visualize similarities and dissimilarities in the gene expression data. So our first hypothesis was a treatment-driven expression model in which treatment explains the vast majority of the data as shown by the separation of genotypes on PC1, and treatment explains a smaller amount of variation in the data as shown by the smaller separation of samples by treatment on PC2. And in comparison to that, we came up with the genotype-driven expression model, in which the exact opposite, where samples are more strongly separated by genotype on PC1, which explains the vast majority of data, and samples are separated less so by treatment, as shown by a smaller separation on PC2, which explains a smaller amount of variation in the data. And then, for a Pastylopora, because we had 8 genotypes, we selected the 3 largest clonal genotypes to test these 2 models, which represented about 85 of the 120 samples that we had collected RNA-seq data from. So, where do our data fit? So, I should say beforehand, we had, of these 3 genotypes, we had 2 triploids and 1 diploid group. And we can see that they seem to appear to fit the genotype-driven expression model, where PC1 explains 24% of the variation in the data, and our 2 ploidy groups are separated on PC2. And as an aside, if you look at the paper, you'll see that when we look only at the 2 triploid groups, they also appear to explain about 28% of the data. And then here, when you look at PC7, it only explains 2% of the data, but it is highly correlated with treatment. And then if we go back to our Montipora capitata, unfortunately, we're not able to test the genotype-driven expression model because we had all distinct genotypes, but we can see whether it fits the treatment-driven expression model. And although we do see some separation on PC2, which is highly correlated with treatment, we cannot say that it fits the treatment-driven expression model because it's not the vast majority of variation that's driven by treatment. So I'm going to skip ahead a little bit because we just heard that second chime. So skipping ahead to our differential expression responses, we were able to detect stress in a single clonal lineage of the triploids, but not within across different genotypes. So what does this all mean? A broadly conserved transcriptome response to stress does not appear to be shared between or across genotypes, which does not support our strongman model of treatment-driven expression. And although we were unable to test by genotype by treatment effects because of limitations in our experimental data set, that is testing for genotype after the fact, it does seem that genotype and ploidy appear to drive genome-wide response to stress, and genotype by treatment effects likely drive these interactions. We think that gene expression may reflect a past adaptive response to local conditions. That is, these genotypes in Pastylopora acuta picked a lane, and because they may end up anywhere, they a bet hedging strategy in which maybe one of them will survive if they land in a habitat where that happens to be suitable for them. And we think that post-transcriptional regulatory mechanisms may be an important process to modulate the gene expression to the physiological responses that we see under stress that underlie the bleaching response. And these results underline the importance of characterizing the genetic structure and reproductive behavior of corals or any other organism before you perform these experiments. So, some outstanding questions. 60% of the Hausloper acuta in Kaneohe Bay are triploid. What makes them so successful? How prevalent is polyploidy in corals overall? And what is the fate of polyploids under increasing anthropogenic stress and climate change, given their inability for genetic recombination? And with that, I'd like to thank my funders, and if you're interested in learning more, please see the following publications.